Alright guys, early in the previous session you learned the significance of meiosis and genetic diversity by looking at independent assortment, um, crossing over, and you looked at dihybrid problems. And what I'm going to do in this session is I'm going to show you a quick, easier way to do dihybrid problems. If you look at step 1, the first thing you're going to do when you're on your test is that you're going to read the genetic problem. In pea plants, green pod color G is dominant over yellow pod color lowercase g. Tall plant height capital T is dominant over short plant height lowercase t. A tall green pea plant, homozygous for height, heterozygous for pod color, is crossed with a tall green pea plant, heterozygous for both traits. What is the probability that we have an offspring that is homozygous dominant for both traits? So, the first thing you're going to do, guys, you're going to read the problem and you're going to highlight the key information. Or you're going to point out what the key information is. For example, if I was reading this problem, the first thing I would do is that I would go ahead and write my alleles for each trait. I have capital G for dominant, and that's going to give me green pod color. And lowercase g for yellow, which is going to give me the yellow pod color. Capital T for tall, because that's the tall plant, and lowercase t for short plant. Once I have my alleles, I pulled out the key information or some of the key information out of the problem. So the next thing I'm going to do is that I'm going to write the genotype for each parent. Remember, the genotype is represented by the letters. So now you got to remember your vocabulary. So let's look at it. It says a tall green plant homozygous for height. Remember, the term homozygous means I have the same allele. So that's why here I have a capital T, because it said it was tall. And it's heterozygous for pod color. Heterozygous, I mean the alleles are different. So that's why I have a big G, lowercase g. It said I'm crossing with a tall green pea plant. Tall, that means if it's tall, and it said it was heterozygous for both traits, it's homozygous dominant. It's crossed with a tall green pea plant, heterozygous for both traits. So heterozygous means that the alleles are going to be different. So that's why I have a big T, lowercase t, a big G here, lowercase g here. Now, since this is a dihybrid cross, remember the prefix di means two. So I'm looking at two traits. That's the same thing as essentially doing two monohybrid crosses. So the first trait I'm going to look at is going to be height. So I'm going to get the alleles from parent 1, which is big T, the alleles from parent 2, where big T is little t, let me go back. There we go. Little t, big T, little t, and then I'm just going to do a normal Punnett square. So I've only looked at one trait, and that one trait was height. So now I'm going to do the same thing for pod color. I'm going to look at these alleles with a heterozygous big G, little g, put those here. This is also heterozygous big G, little g, so I'm going to put those on the side. So then I'm going to cross those. So again, once I got my genotype, I just split them up and I just looked at one trait and did two monohybrid crosses. Now once I do my crosses, I'm looking at a particular genotype. It says, what is the probability that we will have an offspring that is homozygous dominant for both traits? So I'm looking for big T, big G, big G. So my hint is that I got to determine the amount of each desired genotype for each Punnett square, and then I'm going to multiply. And the reason I'm multiplying because there are two independent events. Okay? So let's look at big T, big T. If I look at big T here, I have 2 out of 4. So that's why I put 2 fourths here. Here, I'm looking for big G. So I have 1 here, 1 out of 4, so that's how I have 1/4 there. So when I have 2/4 times 1/4, so now that gives me 2/16. I can reduce that to 1/8, or if I could put it in percent, 12.5%. So let's recap this to make sure we're all on the same page quickly. The first thing you're going to do, you're going to read the genetic problem. Then you're going to write the alleles for each trait. you're going to determine the genotype of each parent. So when I read this, remember, I had homozygous for height, so that's why I got the two big T's. Heterozygous for pod color, so that's why I got this genotype. This one said they were heterozygous for both traits, so that's why I have big T, little g, big G, little g. Next thing I did is that then I did two independent Punnett squares. I broke that dihybrid to two monohybrid. I looked at one trait. In this case, I looked at the height. In this case, I looked for the pod color. So I just looked at those, and I got what my possible outcomes of genetic recombinations are going to be. Next, I just looked at what's going to be, I wrote my genotype for what my offspring is supposed to look like. So it said if the offspring is homozygous for dominant for both traits, that means big T, big T, big G, big G. Now I want to count how many of each one of those I might have. Big T, I had 2 out of 4. Big G, I had 1 out of 4. Because those two events happen independently of one another, I had to multiply. So 2/4 times 1/4 is 2/16. I can reduce that to 1/8, or I can divide 1 by 8 and give 0.125, or I can turn that into percent. I hope this helps.